Hello, everybody. Today I want to talk to you about gene expression in the wire-tail mannequin brain. But before I do, I want to say thank you to my collaborators, all of which have played an integral role in the Mannequin Project. And without their assistance and big brains, the work that I've been able to do certainly wouldn't happen. And the work that I'm presenting today is the result of a true collaboration. And I also want to say thank you to the Mannequin Genomics RCN for providing me with opportunity to interact with some really talented scientists from across the world, all of which have a passion for mannequins, and it's been a wonderful experience, and I'm grateful for that opportunity. Wire-tailed mannequins are an interesting social system because males engage in a cooperative courtship display to impress females. And these display partnerships are long-term, and they form the basis of a social network. And what's important is that a male's connectivity in this social network predicts his fitness. What I mean by that is males with a higher degree of social connectivity, that is a higher number of display partners, tend to have higher reproductive success. We learned some time ago that testosterone may be important in regulating variation and behavioral phenotype. We learned first that territorial males have higher testosterone levels than do floater males. And subsequently, more recently, we've learned that the relationship between testosterone and social behavior is status dependent. That is, the manner in which variation in testosterone predicts variation in behavior is different for territorial males than it is for floater males. I strongly encourage you to check out Rosalind Dakin's talk because she'll go into this discovery in much more detail and will teach you a lot more about the social system of these animals. Now, the overarching goal of our Mannequin Project is to better understand the neuroendocrine and genetic basis of individual variation in male social behavior. But in order to get to this really big problem, we had to do a couple of things first that I'll focus on today. First, we had to be able to quantify gene expression in behaviorally relevant regions of the mannequin brain. Second, we had to learn more about broad and fine-scale patterns of gene expression across nodes of what we call the social behavior network. And I'll explain what that is. The vertebrate social behavior network is a series of reciprocally interconnected nuclei across the brain. The reason they're important to us is because these nodes or nuclei of this behavioral circuit have been tightly linked to the regulation of a variety of social behaviors. And this neural network has been highly conserved. It is evident in virtually all vertebrates studied to date. Don't worry too much about the names of the various nuclei right now, but what I want you to appreciate is that this interconnectivity that they have means that they talk to each other. And collectively, these various nuclei regulate social behavior. That what we know is that not one particular nuclei of the brain is responsible for a particular behavior, but rather activity across this behavioral circuit is going to be responsible for influencing variation in behavior. These different nodes or nuclei of the social behavior network come from different parts of the brain. Nucleus tenea is widely recognized as the homologue of the mammalian medial amygdala for birds. The bed nucleus of the stria terminalis is recognized as the extended amygdala. One node comes from the septal region of the brain. This is the lateral septum. One node comes from the midbrain. The most recognized region is midbrain central gray. And three nodes come from the hypothalamus. That is, three of the nuclei in this network are hypothalamic. And these are the medial preoptic area, the anterior hypothalamus, and the ventromedial hypothalamus. Most of these nodes are a portion of what's known as the basal or limbic forebrain. And then, of course, two nuclei as part of this network are midbrain regions. In order to sample these nuclei of the social behavior network, we collected brains from males that were first characterized for behavior and social status in the field. that's one thing that makes our neuroscience, if you will, relatively unique in that our animals are well understood before they're collected. We're very selective in the animals that we collect. The brains were harvested very soon after capture and frozen on powdered dry ice in the jungle. And then we transferred these frozen brains into what's called a dry shipper for long-term storage and transportation back to the United States. And a dry shipper is a canister that has a unique absorbent matrix that will absorb liquid nitrogen, and it can keep tissues frozen at minus 100 degrees Celsius or colder for 21 days or more. So it really allowed us to store our tissues safely in the jungle and to get them back to the United States frozen. In order to get high quality RNA, it was important that our tissue stayed frozen from the time of collection until the time the tissue was put into trizol as the first step of RNA extraction. So we cryosectioned frozen brains into 150 micrometer coronal sections. And then those sections were put on frozen slides, and we located particular nuclei of the social behavior network using well-known landmarks in the brain. And those, a circular portion of those nuclei was excised using what we call a micro punch or a tissue punch. And we collected samples depending on the size of the nuclei that ranged from 300 micrometers in diameter to a millimeter in diameter. And those little tissue punches were then put into RNA extraction, and we extracted RNA, and we were able to acquire very high quality RNA. On average, our RNA integrity number was 8.95. We did RNA-seq for each brain region of interest for each bird. Ultimately, we collected tissue samples and extracted RNA from 10 different regions from 16 birds. And so this was a lot of RNA-seq, if you will. For our first wave of the work, we originally mapped our reads to the Mannequin vitellinus genome, but thanks to the work of a host of RCN members, we were able to eventually, for our current work, we now map our RNA-seq reads to the Pipra filicauda or wire-tailed mannequin genome. Now, one of the things that we realized from this work is that there is a region-specific pattern of gene expression across nodes of the social behavior network. The first thing we did is we looked at how overall gene expression profiles cluster using a principal components analysis. And this revealed a striking pattern. What we found was that the different nuclei show very different gene expression profiles. We see some clustering of these profiles based on brain anatomy, where they come from. So for example, midbrain central gray and the intercollicular nucleus are close together probably because they're anatomically related and functionally related. But you can still see that their gene expression patterns are distinctive. The anterior hypothalamus is quite distinctive than more ventral portions of the hypothalamus. But even within this neighboring regions of the hypothalamus, we still see distinctiveness, for example, in the paraventricular nucleus, medial preoptic area, and the ventromedial hypothalamus. The bed nucleus of the stria terminalis, which is extended medial amygdala, clusters very differently than nucleus taenia, which is recognized as more medial amygdala. And our one node from the septal region of the brain has a very distinctive gene expression profile. To put this clustering in context, here I've overlaid the major subdivisions of the brain to point out that although we see clustering according to the larger region of the brain that these nuclei come from, we still see distinctiveness within these major subdivisions. Another way to look at variation in gene expression profiles is to do a weighted gene co-expression network analysis. And if we look at modules or suites of co-expressed genes, we see distinctive expression patterns as we look across the various nodes of the social behavior network. Let me show you a couple of examples. If we look at the first three nodes, in this figure, we can see that in 2 of the amygdala regions, genes in these first 3 modules are upregulated. But if we look in the lateral septum, these same genes in these modules are strongly downregulated. As another example, if we look at genes within these 2 modules right here, these are downregulated in the amygdala tissues and the amygdala nuclei, and upregulated in hypothalamic nuclei, the medial preoptic area and the ventromedial hypothalamus. One of the things that we're most interested in is variation in the expression of neuroendocrine genes. And interestingly, most of the neuroendocrine genes we're interested in were assigned to just 4 modules. If we look within those modules, we can see that there is some variation in the expression of genes across the nuclei of the social behavior network, but we also see great similarity within modules. For example, you can see the similarity and patterns of expression for vasoactive intestinal peptide and androgen receptor here, but even within a module, we see differences. So here is the lateral septum. The expression for androgen receptor in the lateral septum is very different than VIP expression in the lateral septum. Perhaps more strikingly, as we look across modules, we see very distinctive expression profiles for the particular genes within that module. As an example here, if we compare this upward trend as we move from left to right across nodes of the social behavior network, we see increasing expression for VIP and AR, androgen receptor. But if we look at, for example, serotonin receptor in a different module, we see decreasing expression as we move across the nuclei. So to conclude, patterns of gene expression across the social behavior network reveal region-specific variation in neural activity. What this means for those of us who want to understand the neuroendocrine and genetic basis of social behavior is we need nuclei level resolution. Whole brain approaches are going to tell us very little about how variation of gene expression in the brain predicts behavior. And looking at larger subdivisions like the hypothalamus compared to the amygdala may not tell us enough either, that we need to get down to nuclei level resolution because of the heterogeneity in gene expression shown here. Thank you for your time, and I strongly encourage you to go watch Perry Bolton's talk to learn more about the next wave of this research.